Thank you for having me. I would like to preface by giving credit to my wonderful collaborators, Dr. Robert Baiocchi from The Ohio State University and Dr. Michael Jordan from Cincinnati Children's for working together with my group on this project. It's a pleasure to have worked with your organization over the past more than a year by now. And the work was supported in a large part by you guys, and I loved going to the meeting this past November and meeting everyone in person, or many people in person, so it's really been excellent. So to tell you a bit about me, so I'm an assistant professor at the hematology division at The Ohio State University in Columbus, Ohio. I did my undergraduate at Vanderbilt, then got my MD and PhD degrees at Dartmouth, and then I came to the Ohio State for my hematology oncology fellowship. So I got my training as a cancer doctor here, and then I stayed here as a physician. I specialize in treating patients with hematologic malignancy, so blood cancers, especially those patients that need therapy with either a bone marrow transplant or cellular therapy. I also see patients with secondary HLH. So that's HLH triggered by another condition. In particular, those triggered by lymphoma. In fact, I had a patient like this just last week. So we know that in adults, HLH can be triggered by other conditions like an infection or an autoimmune disease, for example, autoimmune arthritis, or a cancer such as a blood cancer lymphoma. All of those conditions can trigger this excessive inflammation that results in the syndrome of HLH. As I encounter these patients more and more and read the literature about them, our audience knows, and you guys know, that condition, HLH, can be really hard to treat. And the mortality is high. And the drugs available to us and approaches available to us are limited. So that in particular made me and my colleagues interested in learning more about how to approach this disease. And in addition to working as a doctor, I have a laboratory for translational research. So several years ago, we decided to try targeting and studying this disease in a mouse model. So we replicate the inflammation of HLH that's seen in humans. We replicate it in mice and then try different drugs to treat this inflammation. And we had a particular drug that we've been working with for a while, an inhibitor of this protein that you mentioned, called protein arginine methyltransferase 5, or PRMT5. And from our previous work, my colleagues' work, and also work of other laboratories throughout the country and in other countries as well, there have been data showing that this PRMT5 likely contributes to inflammation in various scenarios. So inflammation of autoimmune disease, inflammation of graft versus host disease, and we also saw in other systems that it modifies immune response. And we wanted to test what if it is also participating in HLH. That's what we tried. And amazingly, from our first experiment, we saw that it looks like if we induce HLH in the mice and then treat them with this drug that blocks PRMT5, a lot of the signs of inflammation go away. So for example, these mice, when they develop HLH, they lose a lot of weight. And the weight loss got a bit smaller when they were treated. These mice get enlarged organs. So they get an enlarged spleen and enlarged liver. And if we treat them, these signs went away. In the liver and in the spleen, they develop an accumulation of these effector cells that go ahead and damage their tissues. The expansion of these cells reduced. And furthermore, these mice develop a very high level of interferon gamma in their blood So that is a cytokine or immune molecule that promotes inflammation. And if we treat them with this drug, the PRMT5 inhibitor, the level of this interferon was suppressed. To us, that seemed really promising, and we wanted to develop it further. And we worked on verifying these results and developing these results probably for 2.5 years to 3 years overall. And finally, we got it to the stage of the publication. So I'm very glad that this finally came out. Right. So indeed, we saw some additional findings. So the level of this PRMT5 protein increases in immune cells of mice that develop HLH. And that also points to an active pro-inflammatory role of this protein in the disease pathology. But we still don't know what exactly it does. This protein attaches methyl groups to arginines of other proteins, and that affects multiple types of cellular function, like basic cellular function, from reading genes, copying genes, making new proteins, or protein to protein signaling. All of those functions have been demonstrated as PRMT5 dependent in various scenarios, so in different cancers, in different inflammatory processes. What exactly is going on with PRMT5 activity and what we change with PRMT5 inhibition that leads to suppression of inflammation in our system, we still don't know that. So there is a lot of work that still needs to be done to understand, first of all, what is PRMT5 exactly doing in inflammation of HLH? And when we block it, how do we prevent this inflammation? So we have some hypotheses about what's happening. So some of our preliminary experiments showed that interferon production may be affected, whether it's affected directly or indirectly at the level of protein production or epigenetic mechanisms, we still don't know. So we have ongoing experiments right now to try to understand this. So we're trying to look at whether the reading of the interferon gene and production of interferon protein is affected, or if other intracellular processes are changed with induction of PRMT5 expression and inhibition of PRMT5 activity. So we're looking at that, and we're seeking NIH level funding for this work to take this further. And it is important to understand how this is acting in order to develop the best approaches that we can translate to a clinical trial in patients. So that's one thing that we're pursuing. Another thing, we're also testing this PRMT5 activity and PRMT5 inhibition in various HLH scenarios. So what I described to you up to now is secondary HLH, i.e. HLH that's induced by another condition. But humans also get primary HLH. That's usually diagnosed in children and babies, and it can be also really severe. So working together with Mike Jordan and my colleague Rob Baiocchi at Ohio State, and Mike Jordan is in Cincinnati Children's, we're trying to assess what happens with PRMT5 and blocking PRMT5 and models of primary HLH also in mice. Those are two of our directions right now. And eventually, the goal is to translate this to patients. So right now, our studies are in mice, but the reason we're really doing it is not just out of interest, but also so that we can help our patients. Because honestly, as a physician, the worst scenario to be in for a doctor is if you have a patient that's really sick and you have nothing else to give them. I want to never be in that position. So ultimately, the goal of all of this work is to find something that we can give to our patients and it'll help them. We are hoping to translate our results from the bench to the patient. We're working towards developing a clinical trial using PRMT5 inhibitors alone or potentially combined with other drugs in patients with HLH. So that's the ultimate goal. In fact, sometimes we see some of these mutations that are present in kids with primary HLH. Sometimes we see them in adults with secondary HLH. But each of us has two genes for every function, one from our mother, one from our father. And in children with HLH, they're usually both affected. But sometimes we see only one gene affected in our adult population. Not every time, but sometimes we see it. So I think there are many ways in which these conditions can be related, absolutely. Certainly, with the support of your society, the work has gained momentum. We certainly, aren't letting it go at this stage. We're absolutely pushing forward and hoping for this to culminate in a clinical trial within 1 or 2 years. That's the hope. We have tested a PRMT5 inhibitor in an all-comers resistant cancer patients. So it was tolerated well. We didn't see a lot of improvement of cancer in that particular scenario, but the toxicity profile was also completely acceptable. So that suggests that the drug can be used in humans, which was very good news. And we're going to use a lot of the data gained from that experience to build our future clinical trial for the HLH patients. Absolutely. Thank you very much.